Next, we're going to go ahead and take a look at the 3 stages of cell signaling. So remember, you've got signal reception, you've got signal transduction for stage 2, and then you have stage 3, which is the cellular response. So let's go ahead and start with stage 1, which is signal reception. This is where the signaling molecule will actually bind to the receptor protein on the target cell. When this happens, that receiving receptor will end up changing shape, typically. So the way that this happens, if we take a look at it, we've got our signaling molecule down here, and we've got our purple receptor protein that's, in this case, embedded in the membrane. The signaling molecule, we can call this the ligand or the ligand. It's pronounced both ways. The ligand will bind to the receptor, and again, this interaction between the ligand and the receptor, the signaling molecule and the receptor, is very specific. So an insulin molecule will only bind to the insulin receptor. It's not going to bind to any other receptor that might be hanging out on the surface of cells. And again, this all comes down to molecular shape. The receptor proteins have that very specific binding pocket that will only allow them really to interact with that one type of signaling molecule. And then once you have that binding occur between the ligand and the receptor very frequently will then change shape and elicit the second stage of cell signaling, which will be signal transduction. We'll get to that here in a second. But because these receptors are so important, we definitely need to talk about them a little bit more deeply. Not all of the receptors are built or function the same. There are 2 main categories of receptors that can be used for cell signaling. Some of them are going to be plasma membrane receptors that are embedded in the plasma membrane or the cell membrane. These are most common. But there are some receptors that are actually found inside the cell, and we call these intracellular receptors. And we're going to take a look at both. Let's go ahead and start with our plasma membrane receptors. There are 3 main types of plasma membrane receptors that we're going to take a closer look at. These are, number 1, our G protein coupled receptors, or GPCRs for short. Number 2, we've got our receptor tyrosine kinases, or RTKs. And then number 3, more specific, we've got ion channel receptors. These are most frequently going to be used as part of synaptic signaling. So we'll take a closer look at those. Now, when we're looking at plasma membrane receptors, keep in mind that these guys are going to be bound to the membrane. They are going to be transmembrane proteins. They have to peek out on the outside the cell, they have to span all the way across the membrane, and then they have to peek into the inside of the cell as well. And so if these transmembrane, membrane-associated proteins are going to function as a receptor, really the kinds of signaling molecules that they're going to interact with are just going to stay on the outside of the cell, and they're mostly going to be water soluble. They're going to come and diffuse through the extracellular fluid to reach the receptors that are hanging out there on the plasma membrane. So let's take a closer look at that first category of membrane receptors, these GPCRs. So this is a complex pathway that starts the signaling process. There are a lot of different kinds of proteins involved here through the use of a G protein coupled receptor. So let's pick this apart. The name of the receptor is a G protein coupled receptor. So we know this is a receptor, and it's coupled to something that's called a G protein. So let's take a look at what a G protein is. And the first panel here, this teal looking protein, this is our G protein. Now G protein is called a G protein because it has the ability to interact with a molecule that's called GDP or GTP. This is the second cousin of ATP. Remember, ATP is this energy-rich molecule that's used for energy processing in the cell, and it's an adenine nitrogenous base with 3 phosphates attached to it. Well, GDP and GTP are the same thing, except instead of an adenine, you have a guanine nitrogenous base. And then the TP is the version with 3 phosphates. The DP is the version with two phosphates, right? Diphosphate versus triphosphate. So the G protein itself has the ability to interact with this GTP or GDP molecule. When it interacts with the triphosphate version of the GTP, this is the higher energy version of the molecule, then the G protein is active. When the G protein, on the other hand, is interacting with the GDP, only two phosphates in that molecule, this is a lower energy version of the molecule, and the G protein interacting with the GDP will be inactive. So that's one part, the G protein. Now, the G protein has the ability to interact with our GPCR. So this is our G protein coupled receptor. This is the protein that will actually interact with the signaling molecule, and you can see that here. Once the signaling molecule binds to the G protein coupled receptor, the receptor is going to change shape, and it's going to allow it then to interact with the G protein. Now the G proteins, they're kind of surfing the inside surface of the plasma membrane. They're mobile, right? They can move across the surface internally of the plasma membrane. And so if this inactive G protein attached to the GDP accidentally maybe bumps into one of these GPCRs that has a signaling molecule bound to it and is in its active state, then the receptor will encourage the G protein to swap out its GDP for a fresh, new, high-energy GTP. And once that happens, you'll notice this G protein is now glowing. It's in its active state because it's interacting with the GTP. And because it can move, this now activated G protein will leave the receptor entirely and float off on the internal surface of the membrane. And if it bumps into an inactive enzyme that it can recognize, it'll activate the enzyme. And so you can see that happening here. You'll notice that the G protein now bound to the GTP left the receptor, migrated across the surface, internal surface of the membrane, bumped into this enzyme and activated this enzyme. This enzyme is then responsible for eliciting the cellular response. Once you elicit that cellular response, you kind of want to stop the whole signaling process because very frequently if you don't shut things off in a cell, bad things will happen. So in order to shut off the signaling process here at the very beginning in stage 1, essentially what will happen once the G protein has left the receptor, the signaling molecule will come off, so you can see that happening here, and the receptor is no longer activated, so it kind of goes into slumbering state. Also, after the G protein has activated the enzyme, it's like, okay, I did my job, I'm done. And so it will be able to hydrolyze the GTP. That means it'll cut off that third phosphate of its own GTP, release it, and what's left will hang on to the G protein, and what's left is that GDP. And remember, any G protein that's hanging out with the GDP, the lower energy molecule, that will be an inactive G protein. So we've kind of returned everything to its inactive state, including the enzyme. Once the G protein detaches itself from the enzyme goes back to its inactive state. And so everything has kind of been returned to that original position, and it's waiting for a new signaling molecule to come along and bind to the GPCR to activate this process one more time. So that's our GPCR. Let's then take a look at our RTKs, or receptor tyrosine kinases. These guys function in a slightly different fashion. So again, it's a complex term, and it's a complex receptor as well. Let's pick it apart. So we've got a receptor, and it also happens to be a tyrosine kinase. Well, if you've never heard the term kinase before, you'd look at the ending, you notice it's an ASE ending, and immediately you're going to think it's an enzyme, right? Well, kinases can function in the process of phosphorylation. These are enzymes, and their whole goal is to phosphorylate other substances, meaning attach a phosphate group to it. And specifically, if it's a tyrosine kinase, then this kinase prefers to phosphorylate the amino acid tyrosine on other proteins. And it's a receptor. Okay, so let's take a look at how this guy functions. Receptor tyrosine kinases are one example of a protein that reaches that quaternary level of protein folding. It's made out of more than one polypeptide. It actually exists as two polypeptides, and each one folds and forms a subunit. So you've got one subunit here and another subunit here of the same RTK. And when they are not activated, meaning a signaling molecule is not bound to them, they pretend that they don't know each other and they're completely separate within the membrane, right? So they're pretending they're strangers. But as soon as a signaling molecule comes along and docks to both of the subunits of the receptor tyrosine kinase, then all of a sudden they remember that they like each other. And the two will form what is called a dimer. Essentially, that just means that they come together on the membrane and they bind to each other. Now you've got an activated receptor tyrosine kinase. And what will happen next is, very kindly, one of these subunits says, hey, buddy, let me phosphorylate you. And the kinase, the enzyme part of the receptor, will then take 3 ATP molecules, take the last phosphate group off the ATP, and instead attach it to each one of these 3 tyrosine residues on its other subunit, so on its buddy, right? So the guy on the right will phosphorylate the guy on the left. And then the guy on the left is like, wow, that was so nice. Let me return the favor. And the subunit on the left will phosphorylate the subunit on the right until both subunits have used up in total 6 ATPs to attach 6 phosphate groups, 3 on one subunit, 3 on the other subunit, to those tyrosine amino acids in the tails of the receptor subunits. Once this happens, now the receptor not only was activated by the signaling molecule, but is also activated through the phosphorylation, and that phosphorylation of those subunit tails allows the receptor tyrosine kinase to interact with relay proteins on the inside of the cell. So these are molecules that are going to come along. They're going to interact with the phosphate groups and say, oh my goodness, I am now active, and they're going to go off and do some sort of cellular response. And the cool thing about RTKs, because of all of these phosphate groups, the RTKs can actually interact with a variety of different relay proteins. So they have the ability to, at the same time, activate multiple cellular responses. So this makes things pretty efficient, right? If the cell says, I really need to do all these things, it'll send that one signaling molecule. And if that one signaling molecule is paired with an RTK, once that RTK is active, then a whole bunch of different cellular responses can all be activated at the same time. If we go back to our GPCR, GPCRs are not good for that. Usually GPCRs are a one-to-one type of deal. You've got a one signaling molecule that's going to bind, and that's going to activate one enzyme that has only one cellular response that it can start. But RTKs, on the other hand, are much more diverse, variable, I should say, right? You can have that one signaling molecule that all of a sudden can activate a variety of different responses in the cell.